CIBERNED YouTube channel. If everyone is interested in looking at it, or someone cannot attend in your lab or in your group and want to attend the webinar. As in every occasion, let me just remind you that we have this webinar series every other week on Thursdays, usually at 3:30 p.m. If you're interested in future webinars, you can double check the program for the next few months in the CIBERNED webpage, where we have already listed every seminar from now until July. As in every occasion, the webinar will last approximately 30 to 40 minutes, and then we'll have some time for discussion. So if anyone is interested in making a question, please use the chat option and send your name and affiliation, and I will give you the floor to make the question yourself directly to our speaker. Also, I would like to ask everyone to kindly turn your microphones off, and ideally also the cameras off, so we can have a smooth streaming of the presentation. Having said that, I will just go ahead and with the introduction of our speaker today. It is my pleasure to introduce everyone to Ledia Hernández. Ledia is PhD biology at the UCM with a thesis in neuroscience, and then was granted a postdoctoral Fulbright scholarship to join Anne Graybill lab at the MIT, Cambridge, Massachusetts, where she studied electrophysiology of the basal ganglia and behavioral learning in Parkinson disease animal models. After that, she got competitive funding from the Parkinson Disease Foundation and the National Parkinson's Foundation to continue her research, focusing on the molecular mechanisms of dyskinesia. After 5 years at MIT, she returned to Spain to join Professor José Obeso in Pamplona and established the Rodin Group when they moved to the HM-CINAC Center in Móstoles, here in Madrid. She will present today the last results obtained from the optogenetic manipulation in rodents in order to unveil the role of the striatal pathways involved in the generation of dyskinesias and wanted motor features related to the chronic treatment with L-DOPA, the gold standard treatment for Parkinson's disease. So thank you very much, Ledia, for being here, for your willingness to share with us your work. And whenever you're ready, please just go ahead. Thank you.
Thank you, Miguel, for the introduction and for the invitation to be part of this webinar series. And so as you well described, I'm going to talk about the latest results that we obtained in the lab with optogenetic manipulation in animal models of Parkinson's disease. So as the title said, it's called Optodyskinesias, evoking and inhibiting abnormal movements in a Parkinson's disease model. So first of all, I want to introduce the basal ganglia, which is a group of subcortical nuclei that mainly regulate movement and automatic behavior, but being also associated with the learning and limbic processes. So it's formed by several nuclei that they are depicted here in a lateral drawing that you can see. The caudate and the putamen that are merged as one structure in rodents. It's called the striatum. We will be seeing a lot of that during this presentation. Then there's the globus pallidus, which has like a more internal medial part and the external or more lateral part. The subthalamic nucleus, that's just as the name says, under the thalamus. And the substantia nigra that has two regions. The compacta, that's mainly dopaminergic, and the reticulata, which is a GABAergic area. So all these nuclei are tightly connected, making loops that in case of that dysfunction, what causes is movement-related disorders such as kinesia, bradykinesia, and rigidity. So in this drawing, we can see how all these nuclei are in these tiny loops that I told you. They are very dependent, their function from each other. And they are divided in different circuits based on the areas of each, the regions of each nuclei that are connected, making what we know as the motor circuit, which connects the sensory motor and premotor cortex with the more ventral areas of the thalamus and the dorsal part of the basal ganglia. The associative circuit, which is more related with the dorsolateral prefrontal cortex and the lateral orbitofrontal one, and the limbic and paralimbic cortex that make the limbic circuit that takes part of the more ventral regions of the basal ganglia. So here my focus in this talk is going to be in the striatum, what we call putamen, caudate putamen in the human, the striatum in rodents. And this area, this nuclei is the main input of the basal ganglia. And it's formed by, it's 90%, 95% of the neuronal population that we located here are GABAergic neurons and are subdivided in two populations. They are the neurons, the medium spiny neurons they are called, they are the projection neurons that express dopaminergic receptor, the kind of D1, the D1 dopaminergic receptor, what makes the direct pathway that we call, because it projects, those neurons project directly to the output nuclei of the basal ganglia, which is the substantia nigra reticulata and the globus pallidus, that go back to the thalamus and the cortex. So that's what we call the direct pathway, and it's been always associated with a role in facilitating movement. The other majority population, it's these projection neurons that express the D2 receptor, and it's called the indirect pathway because instead of projecting directly to the output nuclei, it has some relay stations in the globus pallidus and the subthalamic that go back to the thalamus and cortex. So this indirect pathway, it's been also associated with inhibition of the movement. So it is believed, it's been shown so far, that the balance between the activation of these two pathways allows or inhibits the creation of movement. And what is the relationship with basal ganglia and Parkinson disease? So in the Parkinson disease, as a disease that is depicted here, was described in 1817 by James Parkinson on his essay on the shaking palsy, where he defined a series of symptoms that he found in patients in London that showed that physical features, the inability to move, rigidity, and slowness of movement. But it wasn't until 1960 that the biochemical changes that occur in the brain were described. And here I'm showing you, like, this is the basal forebrain of a healthy human subject, where we can see this line of black neurons. Those are the part compacta of the substantia nigra I told you before, that express mainly dopamine. And those neurons project to the putamen and caudate in the human, and in the striatum in the case of rodents. So what happened in Parkinson disease is there's a loss of these ventrolateral neurons of the substantia nigra that provokes like a loss of dopamine in the more posterior part of the putamen. So this is a disease where there's a lack of dopamine, what's causing all these motor features. And it wasn't until 1968 when George Kochias began some trials with a form of L-DOPA, which is a precursor of the synthesis of dopamine. And in 1970, it was approved by the FDA to be as a treatment for Parkinson disease patients. And it worked wonderfully. And in fact, it's still the gold standard therapy for Parkinson disease nowadays. And it can be combined or it can be preceded by dopaminergic agonist, but still DOPA is the drug of choice for this disease. Since Parkinson disease is a neurodegenerative disease, that's a chronic disease and it's a progressive, It takes these patients to take L-DOPA for decades, and that comes with a toll, which is undesired effects, sensitization to the dose, and what is known as the L-DOPA-induced dyskinesias, which are abnormal involuntary movements that inhabilitate the movement and the life of the Parkinson patients. So here I'm showing you a video of a Parkinson's disease patient, very advanced stage on his normal state without any kind of medication. You can see how he has inability to move. He cannot stand up. He has tremor on the thumb, and you can see here the very short steps. It's hard for him even to turn around, and it's instability. You see how he has this characteristic tremor, slowness of movement, rigidity of the upper body. So after this patient takes L-DOPA, like 30 minutes before of it, the movement is totally recovered, walks, the steps are longer. There's no tremor on his hand, on his leg. He's able to follow the directions of the neurologist and perform all the hand movements that he's required to do. But as I said, this is what's called like the honeymoon period of L-DOPA, and after 5-10 years, it depends on the progression of the disease, the age of the patient, but undesired side effects as dyskinesias appear. So here's a video of a very advanced Parkinson's disease patient which suffers incredible dyskinesias due to the L-DOPA treatment. So this can be very inhabilitating in your daily life because you really don't have control of yourself. So there's several risk factors associated with the development of dyskinesia, such as the early disease onset, because as I said, it's a progressive neurodegenerative disease. So the earlier you get the disease, the longer you are going to live with it and more chances of develop dyskinesias and another unwanted side effects. The Parkinson's disease duration is the same. It goes hand in hand with the previous one. And the prolonged treatment with this pulsatile stimulation of L-DOPA, because it's taken as a form of a pill, like 3-4 times a day, and that makes that the concentration of dopamine in blood oscillates during the day, causing these dyskinesias. So here, like going back to our depiction of the basal ganglia loops and the direct and indirect pathway, what happens with Parkinson's disease is we have this loss of dopaminergic neurons in the substantia nigra pars compacta that affects directly the dopaminergic content of the striatum. And what's been shown is that this dopamine depletion causes a hyperactivation of the direct pathway that comes up as a result of slowness of movement or lack of movement, and there's inhibition of the indirect pathway that facilitates the fact that the patient is more slow, and there's an unbalance in the regulation of movement by these two pathways. So this is all happening, we believe, at the striatal level, because it's been described that this lack of dopamine in the striatum makes several changes. So the fact that there's non-physiological dopaminergic stimulation, there's this pulsatile, makes changes on the thermic, this is the corticostriatal synapses of the medium spiny neurons, and that makes changes on the plasticity of these synapse, and the response of the receptors D1 and D2 changes also with this dopamine depletion. So we have an abnormal neuronal substrate here that may be the origin of dyskinesia. And also what supports that striatal origin of dyskinesia is the fact that early in 2000s, there were several groups reporting the effects of the fetal nigral transplants on Parkinson's disease patients. Basically, they perform surgeries on this subject, implanting fetal dopaminergic neurons in order to replace the lack of dopamine that they were suffering. And it wasn't a big surprise, I guess, that after 5 years, 2, 3, 5 years, depending on the study and the patients, this cohort start developing dyskinesias. So it was a failure that proved that the striatal could be the origin of these abnormal motor features. So having all that in mind, we had a general aim, which was to analyze what was the dyskinetic behavior with optogenetics to try to disentangle the role of the medium spiny neurons on this motor alteration and see if we could evoke and inhibit this kind of behavior. So this talk is going to be divided in these 3 small aims. The first was to generate those dyskinesias with optogenetic stimulation in an animal model of PD. Then try to activate, inhibit each pathway separately with optogenetics to see the effects of the activation of each pathway. And then finally try to inhibit these L-DOPA-induced dyskinesias with this selective approach. So first of all, I told you that in the striatum, there's more than 95% of the neurons are projection neurons, these medium spiny neurons that are GABAergic, that express D1 or D2 receptors. But there's also like 2-3% of interneurons that are very different. There's from cholinergic, GABAergic, expressing parvalbumin. There's a lot of interneurons that have a role that cannot be dismissed. So in order to try to manipulate and see what is the specific role of the population of our interest, we needed a very selective tool to do that, because any pharmacological approach, as you see, they share dopaminergic receptors, will affect more interneurons than the ones that we wanted. We just wanted to see what's the effect of the projection neurons. So in order to do that, we took advantage of optogenetics to make this selective manipulation and not interfere with the role of the interneurons. So briefly, if there's anybody, I believe everybody knows what's optogenetics, but just a quick introduction. Optogenetics is a technique that allows like a selective manipulation of a specific neuronal population based on the receptors they express or the area where they are located. So these proteins is a channel, is a membrane channel that gets excited or inhibited by a laser. And what we do is introduce these proteins through a gene that encodes it in a viral vector that is injected in our animal. And if it's a transgenic one, we can put a specific promoter that makes the opsin to be expressed just in the kind of neurons that we want, super specific. In this case, for us would be the GABAergic, the medium spiny neurons. And after the period of expression of the virus, you put a cannula, laser, and after activation with the light, you get a behavioral output. So first, I'm going to show you the results of activating both striatal pathways in an animal model of PD. That would be in the aim of mimicking the L-DOPA, because L-DOPA acts on both striatal populations. But in this case, we will be bypassing the role of the dopaminergic receptor. So to do that, we use rats, Sprague Daly. We made a traditional lesion with 6-hydroxydopamine in the medial forebrain bundle, causing a dopamine depletion in the striatum and the substantia nigra. And then we inject a viral vector with the opsin of our interest, with rhodopsin in this case. And we place a laser cannula laser on top of the injection and secure it with dental cement for further experiments where we will assess the effects of this stimulation on the animal. So here I'm showing what is a coronal view of a slide that has a staining for TH, which is the marker for dopaminergic input. You can see this is a rat that got the 6-hydroxy injection, and we see the totally depleted striatum with the nigra also. And in the next window, in the next, you can see the expression of the channel rhodopsin in fluorescence with the expression of it on the medium spiny neurons. And here you can see where the arrows are, like the cannula placement. They were aimed for the dorsolateral part of the striatum, which we believe it's the area more related with the dyskinesia. So basically what we did was this surgery. We did the 6-hydroxy lesion and the injection of the virus at the same time, and wait 3 weeks for the expression both of the 6-hydroxy toxin and the expression of the virus. An habituation of 1 week of the animal to the laser setup and the place where we were going to perform the experiments. And then we did the optogenetic experiments. So for the optogenetic experiments, we use 2 different protocols of stimulation. We wanted to test 3 minutes of continuous light. We wanted to do like a strong stimulus to make sure that we weren't missing anything. And then we did like a smaller activation with a protocol that had only 10 seconds of light in intervals on 9, making it like a more kind of physiological, not so abrupt stimulation like the continuous light. And then we videotape the behavior of the animals and analyze the dyskinetic behavior, scoring for the axial dystonia. You will see is a particular torsion of the body of the rat, and the limb dyskinesia, which is the repetitive movement of the forelimb that's affected. So for you to see how a dyskinesia looks like in a rat, I show you what a dyskinesia looks in patients. This is how a traditional L-DOPA looks in a PD animal model. So we see this is the forelimb dyskinesia, and the axial is this torsion of the body, that twisting that they exhibit. And this is very characteristic from the typical pharmacological dyskinesias. And we give a rating, about how bad that dyskinesia is. So when we have these dopamine depleted animals and we stimulate it with a laser, this is the kind of behavior that we obtain. You see how once the light turns on, the animal starts twisting his body and exhibits a very similar, if not identical, behavior at the one that we obtain with the traditional L-DOPA-induced dyskinesia. So when we can quantify this scoring for dyskinesia for the protocol A that we call, this is the longer stimulation with continuous light for 3 minutes, we see how we evoke, as you could see on the video, dyskinetic score. The time that the animal was exhibiting dyskinesia was increased and significant different from the control animals that didn't exhibit this kind of behavior, and we quantify that for the forelimb and the axial dyskinesia. And also with the milder stimulus, we got similar effects, where there was a significant increase on this kind of behavior in the dopamine depleted animals. So there was a subset of animals that didn't exhibit dyskinesia the first time that we exposed them to the laser. That was only a small cohort, but it happened. So we decided to give them L-DOPA to see if they were well lesioned, because that can happen. But they were well lesioned, as you see here. When we check for the dyskinesia with L-DOPA administration, these animals had both forelimb and axial dyskinesia very significant. And our surprise was that when we plugged those animals to the laser and stimulate them again, we got an increase on the dyskinetic behavior, showing that the L-DOPA was kind of priming this kind of behavior on these animals that didn't exhibit the dyskinesia the first time that we exposed them to the laser. So that spoke about not only the behavioral, the motor output that we saw on those animals was very similar to the L-DOPA, but there was some molecular pathways probably or circuits that they were using that were the same that we were obtaining with Optodyskinesias. So to validate that, we wanted to look for a specific marker that was described in Morataya lab in 2009. It's the FOSV that it lights up, as you can see here, in a dyskinetic animal, in a mouse that they use in this work, where totally depleted areas here, as of FTH showing, had an increase of the FOSV marker, showing that this is a marker for the L-DOPA-induced dyskinesia. So we wanted to test that in our rats, and it was very nice to show that not only the motor output was identical, but also our Optodyskinesias evoked this increase in FOSV the same way that it was defined with the traditional L-DOPA, kind of validating this model on this aspect. And here, as you can see, the increase FOSV cells in the dopamine depleted side, it's way over the graph compared with the control animals that didn't exhibit dyskinesia. So the dyskinetic behavior, it's been associated with the activation of the D1, as I told you on a couple of slides before, and with the help of Morataya lab, we wanted to see if the increase that we got in FOSV was specifically in one of the subpopulations, and it was very nice to show that this FOSV marker was colocalized with dynorphin B, which is a marker for the D1 expressing neurons. And as you can see here in the double immuno, the neurons that were labeled with FOSV were also labeled for dynorphin B, validating the previous studies that were published where they give the D1 expressing neurons a more prevalent and relevant role in the dyskinesia generation. So as a summary of this part, we could say that the optogenetic activation of both striatal pathways induces normal involuntary movements that are very similar to those that are evoked by the traditional dopa administration. That the dopamine depletion is required for the development of dyskinesias or optodyskinesias in this rat model. That there's a facilitation of the L-DOPA treatment in the induction of optodyskinesias in the rats that initially didn't have that kind of behavior. And this optodyskinetic behavior increases the expression of a molecular marker associated with the traditional levodopa-induced dyskinesias. So to move further, we wanted to know what was the role of each pathway, because it was in the literature, as I mentioned before, described that the D1 expressing neurons were responsible for the generation of dyskinesia. And there was several papers later that show the role of the direct neurons in that kind of behavior. But also, there was previous older literature showing that, for example, in monkeys that they were administered pharmacologically with an agonist of D2 receptor, they exhibit also dyskinesias, and there was some recent paper using another novel approach as this, the chemogenetic, that show also a role for the D2 receptors. The lab of Morataya also showed that there was changes in the synapses of the D1 and D2 expressing neurons, with changes in the spine density, which points to the fact that these D2 neurons also had changes in them. So they have to be involved or related, if not the generators, for sure suffer the consequences of the dyskinetic behavior. So in order to see what's the role, that's another work showing that there's a small percentage of the D2 neurons, 10% of the D2 neurons are involved on the generation of dyskinesia. So with all that in mind, we wanted to see what was the role of each pathway. If we could evoke dyskinesia, that would be the way to prove the role of each pathway on the generation of dyskinesia in a more direct way, we believe. So for that, we had to move to mice. And we use wild-type mice, but also we use 2 lines of transgenic mice. The D1 Cre mice that will allow the expression of the opsin exclusively in the D1 expressing neurons, in the direct pathway neurons, and the A2 Cre mice that will allow us to express the opsin only in the indirect neurons of the striatum, of the medium spiny neurons of the indirect pathway. So the approach here was identical to the one that we used for the rats. We use the 6-hydroxylesion in the medial forebrain bundle to induce a depletion in the striatum and the substantia nigra, a dopaminergic depletion, and then we injected the channelrhodopsin in the striatum. In the wild-type animals, we will have affected both pathways the same way that we did with the rats. But in the transgenic mice, in the D1 mice, we will find only the channelrhodopsin and the effects of the light stimulation on the direct pathway, and the same for the indirect pathway with the A2A Cre mice. So here I'm showing you another example of the histology of the dopaminergic depletion obtained in the mice. Here, this is the channelrhodopsin expression on the D1 Cre mice, where you can see the expression of the virus of the opsin in the dorsolateral striatum, the substantia nigra reticulata as far as the direct pathway. And here I'm showing you 2 pictures of the expression of the GFP, the channelrhodopsin, in the dorsolateral striatum and in the globus pallidus, proving that this model, the indirect pathway, was working and labeling what was expected. And here I'm showing how the channelrhodopsin gets a nice infection of the whole medium spiny neuron, as you can see in this picture. So the same that we did with the rats, we used different protocols. We did that for the mice too. We had 2 different protocols where we use a continuous light for 10 seconds. That was one of the protocols. And the other protocol, we use the 3 minutes, but we use a frequency of 10 Hertz, which is a more physiological frequency of firing for these neurons. And like in the rats, we analyze the different dyskinetic behaviors. In this case, we did orolingual for the mice too, the axial dystonia and the limb dyskinesia that we use in the mice. So like I show you the dyskinetic behavior in a rat, you will see that's very similar to the one obtained in a mouse. This is the traditional L-DOPA-induced dyskinesia in a mouse that has a dopamine depletion. And after injection, you have this typical behavior with the twisted body, the grooming, the forelimb dyskinesia, and the constant involuntary and controlled movement. And this is what we got when we stimulate both pathways with the laser in a dopamine depleted animal. So we'll see when the light turns on, the animal starts turning, twisting his body, showing like a very similar behavior to the one that we've seen in the previous video with the other mice. And once the light stops, he stops the behavior. It's like a switch. It's very nice. So I'm going to show you the results that we obtain when we stimulate both pathways, like the same experiment that we did in the rat. We wanted to validate it first in the mice and then move to the transgenic. So here, for our surprise, we had the sham group and the dopamine depleted, and you can see here how both animals exhibit dyskinesia when the laser was administered, both in the continuous light or the 10 Hertz stimulation, which was very shocking because we wouldn't expect this from the literature, the knowledge known so far, and for sure for the same results that we obtained in the rat. So that was a very surprising, unexpected result. And these experiments also show us that the continuous light it's a stronger stimuli than the 10 Hz light, where we could see like a significant effect of the light and not so clear on the milder and the 10 Hz, more physiological stimulation. But in both cases, dyskinesias were evoked in control and dopamine-depleted animals. So this is how the stimulation of one pathway, the direct pathway, looks like. We were able to evoke dyskinesias just stimulating the direct pathway. As you can see here, when the light turns on, the animal starts twisting his body, turning around, forelimb akinesia. The light turns off, he stops the behavior. It's very clear. So similar that what we obtained with both pathways here, both in dopamine depleted and control animals, we had the same effect. We got dyskinesia just stimulating the direct pathway on these animals, having scores for dyskinesia in all the categories that we analyzed, both orofacial, axial, and forelimb, both with the continuous light and the 10 Hz light. And as you can see here, the effects of the 10 Hz light were reduced to the first stimulation, and after that, like the effect of the opsin gets desensitized, and we don't have this kinetic behavior anymore. So what we wanted to do also was to record those neurons while we were doing the stimulation, because there's been controversy about the effects of the laser on the population. Sometimes they inhibit the behavior, sometimes they get excited and then they stop responding. So here, what we can show here, this is the firing rate of a subset of dopaminergic neurons that were stimulated with the laser, as is indicated in the graph. And we can see how nicely they increase their firing through the whole time that the laser is given on. So those are the label neurons. And then we have like a subset of neurons that were inhibited, but those were not infected with the opsin. So we believe that those be the neurons that are affected by the activation of the other population. So we wanted to compare also what was the difference, if there was any, between the stimulation of one pathway and both pathways in these dopamine depleted and sham mice. And we could see how the continuous light is, as we saw before, like a stronger stimulus that evokes higher dyskinesia score when the laser is turned on. And also with the both pathway stimulation, we cause like a higher dyskinetic behavior, despite it's not significant. So we could say that both kind of stimulation are able to evoke dyskinesia in these conditions. And as we did with the mice, we wanted to see the effects of the L-DOPA on this stimulation, and we could see how the L-DOPA made a significant increase on the dyskinetic score when the continuous light was applied, both stimulating both pathways, or when we were stimulating only the direct pathway. So here again, we are replicating the data where we show that there must be common mechanisms and circuits targeted by the L-DOPA and by the laser that make the effects of the L-DOPA potentiate the effect of the laser in both kind of stimulations. And the same thing that we did with the rat, we wanted to validate that with the mice mainly, because as I said, like we got dyskinesia in both animals, in the dopamine depleted and the control ones. So we wanted to see what happened with FOSB, with this specific marker for L-DOPA that Morataya lab showed that it was only activated when dopamine depletion was complete. And we were very happy to see that, sorry about that, that in fact, we only got an increase of FOSB in the dopamine depleted side. In the control animals that they exhibit dyskinesia, they didn't have this FOSB increase of these markers. So that gave us a very strong background to support the fact that this specific marker is a dyskinetic marker, but for conditions of dopamine depletion. Like I said, this is the work that's shown that only in completely depleted areas is where FOSB is expressed, and it certifies or validates the data that we published before, showing this increase in FOSB caused by the optodyskinesia. So we thought till this point that dopamine depletion was a fact, a need to develop dyskinesia, at least with the pharmacological treatment. But we could find like 3 papers in all, after 25 years of L-DOPA, we could find only 3 papers where they showed that there were animals and even patients with preservation of the dopaminergic striatum levels, but they exhibit some kind of dyskinetic behavior, showing that there are exceptions, and probably the manipulation that we do with optogenetic only in mice makes some kind of imbalance between these 2 pathways, evoking dyskinesia in a condition that we couldn't find it otherwise. So I show you what happened when we stimulate the direct pathway, and I'm going to show you what happens when we stimulate the indirect pathway. In this case was the A2 Cree mice that express the D2 receptor. So you will see here, when the laser turns on, the animal kind of stops and turns, moves, but there's nothing similar to what we've seen with the activation of both pathways of the direct pathway. So there's no dyskinesia when we stimulate the indirect pathway. Those are the results showing the rotational behavior. That's what we measure in these animals. And we also got in these animals rotation both in the dopamine depleted and in the control animals. In this case, the rotations of the sham animals were significantly higher than the dopamine depleted in both cases, which kind of makes sense because these animals have more dopamine, more ability to move, and the ones that are dopamine depleted have a smaller range of movement, and the amount of total movement is reduced because of the reduced or lack of striatal dopamine content. And here I'm showing the recordings of this population also when they are stimulated with the laser, showing that we are really activating these neurons with the laser, and those effects are related with an activation of the firing of these populations. And the final aim was, of course, to try to inhibit the L-DOPA-induced dyskinesia, having this specific tool that allows us to manipulate the neural substrates. It's of course, a goal to try to inhibit this kind of behavior. So coming back to the loops of the direct indirect pathway, as I told you before, like this lack of dopamine causes an activation of the direct pathway and a reduced activity on the indirect pathway. So if this is the pathological state, in order to go back to a more normal physiological one, our goal would be reduce the activity of this activated pathway by inhibiting its activity, and the other approach could be increasing the pathway activity of the one that is decreased. And with this in mind, those were the approaches that we did. So first, we tried to inhibit the dyskinesias with direct pathway inhibition. And for that, we treated the mice with L-DOPA for 5 days to induce dyskinesias, and then we exposed them to the laser and tried to see if with this approach we could inhibit the dyskinesias evoked by L-DOPA. So this is, as you see, an animal with the traditional L-DOPA-induced dyskinesia. We give them the yellow light. This is the allorodopsin. It's a opsin that inhibits, or supposes to inhibit the neuronal firing of this population. But as you can see, the animal couldn't care less. He keeps going on with his dyskinesia. So we measure the dyskinesia levels like at the 3 measure points where the L-DOPA has effects. There's usually like 60 minutes is the maximum that you check for this behavior after L-DOPA injection, and there was no change on the dyskinesia score. So the other approach would be, instead of inhibiting the direct pathway, to inhibit the indirect pathway. To activate the indirect pathway. And for that, we did the same thing. We primed the animals with L-DOPA to make them dyskinetic. And then the day of the experiment, we plugged them to the laser, study the behavior for that hour. And the stimulation protocol, we use the both that I described before, one with continuous light and the other one with it on Earth, to see which one could work better. So this is one of the videos of the experiment with the animal with the dyskinetic behavior. Once the light turns on, he stops. Starts exploring, looking around, but there's no dyskinesia. It's almost normal behavior for a mice. So there's no spoiler here. Like I show you how the laser reduces the scoring of dyskinesia. You can see here the levels of dyskinesia. When you turn the light on, it dramatically decreased the score for dyskinesia in the protocol where the continuous light was used. And with the discontinuous, with it on Earth, we see how the dyskinesia doesn't decrease significantly till the 60 minutes, which basically could be showing many things. It could be showing that the opsin, the repetitive stimulus of the opsin, is making finally an effect, but also by the 60 minutes, the levels of L-DOPA are reduced. So we have a less amount of dopamine to fight, and probably at this point, the laser can be effective. So since we stopped the dyskinesia, we wanted to see if that has an effect on the FOSB expression. So here I'm showing the FOSB expression for the animals that show dyskinesia and the ones that we stopped the dyskinesia with this approach. And as you can see, we couldn't see a reduction in the FOSB levels, which makes also sense because FOSB is a molecular marker that gets accumulated in the neurons. And we don't expect that by a punctual manipulation approach that stopped dyskinesia, that would reflect in a marker that has such a long prevalence of hours in the cell. So, but that was a fact that we wanted to report to. So kind of to resume the mice experiments, I'm showing you this slide where if this is the motor output from less to more movement, when we have a healthy mouse, we have the balance between the activity of those pathways that allow him the movement that he wants to perform. But when we have a dopamine depletion, would be the Parkinson disease state, there's a higher activation of the indirect pathway that leaves them akinetic and with lack of movement. And if at that point we give them the laser or the L-DOPA to recover their mobility, we evoke the dyskinesia, but we counteract that imbalance. We turn around this balance, favoring the activity of the pathway that releases movement and makes them more hyperactive. So when we do the inhibition with the activation, of the pathway to go back to this equilibrium, we restate the animal behavior, going back to a more normal features of the animals. So from the whole experiments, the set that I show you here today, we can conclude that the optogenetic activation of both striatal pathways replicates the L-DOPA-induced dyskinesia very well. That these optodyskinesias can be induced in dopamine-depleted and controlled mice, while in rats there's need of dopamine depletion. That the selective optogenetic activation of the direct pathway is enough to induce abnormal involuntary movements in both dopamine and non-dopamine depleted mice. That the L-DOPA potentiates the optodyskinesias. That these optodyskinesias share this molecular marker FOSB with the traditional levodopa-induced dyskinesia, but only in cases of dopamine depletion. And that the optogenetic activation of the indirect pathway is sufficient to stop dyskinesias and could be like a therapeutical approach in the future, a way to try to translate that to the clinics and deal with this inhabilitate condition. So these are the funding sources that we all owe our ability to perform the research and the collaborations that we have established to keep moving forward and improve the experiments. So we are located in Móstoles in the HM Center, and all the results that I have shown are part of Ivan Castella's thesis that will be defending his thesis in the coming months. And I want to thank you for your attention.
All right, thank you very much, Ledia, for a nice presentation. So now we have some time for discussion. So as I said earlier, if anyone is interested in making a question, please do so by using the chat option. You don't need to write down your question, just state your name and affiliation, and I will give the floor to anyone interested in the first come, first serve basis. So while we wait for the first questions to come, let me just remind everyone that CIBERNED Webinar Series are held every other week on Thursdays at 3:30 p.m. And if you are interested in checking the future seminars, you can go to the CIBERNED webpage and check every seminar from now until next July, which we already have the program ready for you. So, okay, so we have the first question coming, which is from Mercè Massana from the University of Barcelona. Mercè, just remember to put your microphone on and camera. Please go ahead.
Thank you. Well, first, congratulations for the nice presentation. It's really clear and it's a huge work, which I can understand pretty well. Just to introduce a little bit, so we are also using D1 Cree and A2A Cree mice, in our case in the context of Huntington, but we use similar approaches with optogenetics to stimulate D1, so direct pathway and indirect pathway. And because we were focusing mainly in locomotor activity and motor learning processes, we were not paying attention on the dyskinesias. But I am a little bit familiarized with the movement of the mice that they make this kind of movements. I was wondering, related to your data, if you check locomotor activity or other behaviors when stimulating the direct and indirect pathways in these mice, so that can help also to see how the stimulation unilateral, I guess it was unilateral, how you can distinguish between these dyskinesias, or it's an effect of the locomotion or inducing locomotion with this approach. And then just a second question, then I so that you can answer later. It's regarding the allorodopsin. If you, because I know that sometimes the allorodopsin doesn't work really nicely. If you have a proof that it was actually working on your system, or it could be just a confounding or a technical issue. Thank you.
Sure. Thank you for your questions. For sure, the allorodopsin doesn't work as well as the channelrodopsin. I'm not the only one that says that. It's been already published. It's harder to inhibit neuronal activity than stimulated for some very wise nature reason. And there's more potent. I will recommend you to look for other opsins that work with the blue light. They are recently published. You can email me and I can email you the reference for the new opsins that work with blue light and work way better than allorodopsin. Allorodopsin, it's we didn't record on these neurons, but I can bet part of my money that those neurons weren't really inhibited, because it's really hard to inhibit those neurons with that opsin. Doesn't work as one would like to. And regarding the monitoring of other kind of behavior, like with the D2, we saw that turning that was very clear that that's an increase in locomotor activity, but it's just contralateral turning, so it wasn't really that. And with the other animals, as you could see in the video, there's no locomotion at all. They get stuck in a place, displaying this beautiful plethora of these kinetic movements, but there's no increase in locomotion, no.
Okay, yeah, thank you.
Sure, thank you.
All right, thank you both, Merce and Ledia. Any other questions? I'm not receiving any more questions, but I'd like to give you 1 or 2 minutes just in case someone is still interested. No questions coming through the chat, no hands raised either. So, no more questions. So this is your last chance to ask a question. No? All right, so then I guess we're gonna close the session here. Not before thanking Ledia for a very nice presentation and throwing some light into the issue.
Thank you.
Thank you everyone for attending the webinar. I remind you that the webinar has been recorded, so if you're interested to go back and check anything, you can do so by going through the CIBERNED YouTube channel. We will put the upload the presentation in a couple of days. So thanks again, Ledia, for your willingness to share this nice work with us. Thank you everyone for attending, and we'll have our next webinar in 2 weeks. So have a great day, everyone. Thank you very much.
Thank
Bye-bye.
you. Take care. Bye.